Viral, bacterial, and eukaryotic genomes.
The three parts of a virus are genetic information, the capsid, and the lipid membrane. The genetic layer is made up of nucleic acids. The basic structure of a virus is made up of genetic information and a protein layer called the capsid, which protects the information molecule in the virus. Some viruses have lipid membranes, but not all of them do. Viruses can acquire lipid membranes when they interact with cellular membranes, which causes them to become modified. Viruses cannot live without a host cell, and they use a host cell to reproduce. Viruses inject genetic material into host cells, and those genomes take over the host's organelles, and it kills the host after they assemble.
Hi, this is Ms. Piper, and I am the AP Biology teacher here at Dudley High School. And I am going to be wrapping up and summarizing the lecture on viruses, bacteria, and their genome. And specifically, I am going to be talking about the lysogenic and elliptic cycles. We're going to start with the lysogenic cycle, in which a virus injects its DNA into a bacterial cell, which is its host. And what happens is the viral DNA is integrated into the DNA of the bacteria, and the bacteria goes through cell division, which we call binary fission, and we get two new daughter cells, and those two daughter cells possess the new DNA. When a cell goes under duress or it goes under stress, the DNA is exercised away from the actual bacterial host and can enter in what we call the lytic cycle. In the lytic cycle, the phage or the viral phage attaches to the bacterial cell and injects its DNA, but the DNA remains separate, and then several phages are replicated within the cell. The cell lyses, and then those phages are released, and then those phages are free to take on other bacterial cells as its host. So it's my hope that you've gained an understanding on how bacterial and viral DNA works, and we look forward to hearing from you and speaking with you about it next time.
There are four types of horizontal gene transfer in bacteria: transformation, transduction, conjugation, and transposition. Transformation is the alteration of bacterial cells' genotype and phenotype by the uptake of foreign DNA from the surrounding environment. Transduction is the phages that carry bacterial genes from one host cell to the other. This can happen when the host DNA is accidentally packaged inside of a bacterial capsid instead of the viral genetic material. Conjugation, which is often referred as bacterial sex, is direct transfer of genetic material between two bacterial cells that are temporarily joined through a sex pilus. Transposition is a DNA of a single cell that can undergo recombination due to the transposable elements.
Chromatin is a protein DNA complex. Histones are proteins that help pack DNA into protein. Histone proteins are highly conserved throughout all eukaryotes and are similar to proteins found in prokaryotes. Histone tails interact with each other. They are the most common sites of post-translational modifications. Heterochromatin is tightly packed histones. Tightly packed histones prevent transcription enzymes from accessing the DNA sequence, so these genes are generally not transcribed. Euchromatin is loosely packed histones. Cell differentiation is when cells undergo a process of specialization in form and function. This requires certain genes to be expressed or not expressed. Regulation of gene expression can happen due to regulating chromatin structure, regulating transcription initiation, modifying mRNA, regulating translation initiation, and modifying proteins.